You know, right now, as you're watching this, there's a secret conversation happening inside your head. It's a constant, high-speed chatter between your brain cells that lets you think, learn, and react in the blink of an eye. Today, we are going to eavesdrop on that conversation and find out how it makes your brain so incredibly fast.
So, have you ever stopped to think about that? How does a thought just zip from one side of your brain to the other almost instantly? Well, it turns out the answer isn't just about the nerve cells themselves, but about what's wrapped around them.
And that wrapping is a fatty substance called myelin. Myelin is basically the ultimate biological upgrade for your brain's wiring. It's the secret sauce, the key to that rapid-fire communication between all your neurons. Now, the best way to picture how myelin works is to think about a simple electrical cable. Your nerve fibers or axons are like the copper wire inside. They carry the electrical signal. And myelin? That's the plastic insulation wrapped around the wire. Without that insulation, the signal would just leak out and fizzle. But with it, the signal can leap down the axon at absolutely incredible speeds.
So who are the electricians installing all this high-performance insulation? Well, they're a very special type of brain cell called an oligodendrocyte. That's a mouthful, so we can just call them OLs for short. And these cells are the master builders of myelin, wrapping it around the busiest axons that need that extra speed boost.
But this brings up a really big question, one that scientists have been chasing for years. How does a busy, firing neuron actually signal to a nearby OHL and say, hey, I'm working hard over here, I need some of that myelin insulation? What is that signal?
Well, the first clue they uncovered was the messenger molecule. When a neuron is active, it releases a neurotransmitter called glutamate. Now, for the longest time, we thought this was just for neuron-to-neuron communication, but it turns out the oligodendrocytes are listening in on the conversation. And when an OL hears that glutamate signal, something really dramatic happens inside of it. We see a sudden, sharp spike in calcium ions. This calcium spike is the internal go signal. It's the trigger that tells the cell, okay, it's time to get to work and make some myelin.
So the plot thickens, right? We know glutamate's the messenger, and a calcium spike is the command. But how exactly does glutamate open the floodgates for all that calcium to rush in? As it turns out, it wasn't a simple lock-and-key situation. It was something much more clever. So check this out. To figure it out, scientists ran this really slick experiment. They blocked two possible doorways for calcium, one at a time. And as you can see here, when they blocked the first pathway, the calcium response dropped by almost half. Then, when they blocked the second pathway, it dropped even more. The key takeaway, since blocking just one of them didn't stop the calcium completely, it proved that both of these pathways had to be involved.
And the researchers' own words really nail it. Their results showed that glutamate was activating two totally different types of channels, something called an AMPA receptor and another one called a voltage-gated calcium channel, or a CAV channel. Both were working together to create that one big calcium spike.
And this is the big reveal, the absolutely genius part of this whole system. It turns out that one single trigger, that glutamate, is responsible for opening two different calcium floodgates, one right after the other. It's like having a master key that can unlock two separate doors, but in a very specific order.
Okay, let's walk through this brilliant 1-2 punch. First, an active neuron releases glutamate. That glutamate then latches onto an AMPA receptor on the oligodendrocyte. This opens the first gate, which lets in just a small trickle of calcium. But, and this is the critical part, that little trickle of positive calcium ions changes the electrical charge inside the cell. And that electrical change is the signal that unlocks the second set of gates, those voltage-gated channels, which then swing wide open, letting a much, much bigger wave of calcium flood into the cell. And you might be thinking, okay, that's cool in a petri dish, but does this happen in a real brain? The answer is a resounding yes. The scientists confirmed it. They stimulated actual living axons and saw the exact same two-pathway response in the oligodendrocytes nearby. This is really how the brain works. So, why does digging into this secret conversation matter so much? Well, because this fundamental process is tied to everything from how our brains grow and learn to how we might one day treat some really devastating neurological diseases.
Honestly, this discovery is a huge deal. It helps explain brain plasticity. You know, the whole use it or lose it idea. The pathways you use the most get stronger and faster because they're literally getting more insulation. It gives researchers brand new targets for therapies to help people with diseases like MS, where this vital myelin is lost. But maybe most importantly, it just reminds us that the brain isn't simply neurons talking to other neurons. It's this incredibly complex ecosystem where all the cells are in constant, active communication.
And that just leaves us with a final, pretty mind-blowing thought. If we just uncovered this incredibly elegant conversation, well, it makes you wonder, doesn't it? What other secret conversations are our brain cells having right now, just waiting for us to figure out how to listen in?